Hi, good morning. So I'm Markita Landry. I'm an assistant professor in the Department of Chemical and Biomolecular Engineering. And I want to bring up a topic for discussion within the scope of this forum that is, I think, one of many approaches that we need to take in moving forward with agricultural sustainability. As has been discussed already, we will need to increase our global food supply and also the nutritional value of this food supply in the coming decades. And there are, of course, many different approaches that we can take to accomplishing this goal. Now, specifically, one area that I think is promising to combine into these efforts is in the genetic modification of our plants and our crops. The United States as a whole is a global leader in the production of GM crops, as are listed here. And while many of these crops are used for animal feed, I think the advent of genome editing technologies presents an enticing opportunity to further expand our efforts in this space. Another common example that I like to highlight is the production of golden rice, which was first scientifically reported a few decades ago and has begun gaining some adoption in countries where this varietal of rice could be most useful, in my opinion. Specifically, this is a strain of rice that has had the gene for expression of vitamin A engineered into its genome and therefore constitutively expresses this nutrient that is in need in many populations that are otherwise deficient for it. Now, if we think about both the time and financial effort that it takes to produce a genetically modified crop, we can think a little bit about what is limiting advances in this field. And the limitations are not just in terms of cost and time, which are often interchangeable, but also in terms of the regulatory burden that these crops need to undergo, and also the diversity in the regulatory burden depending on where it is geographically that we're talking about. So today, a GM crop takes about 13 years of R&D at a cost of nearly $146 million to produce. About 40 million of these $140 million are regulatory. So one of the goals of my research group is to first understand molecular transport in plants and then to leverage this knowledge for the development of tools that would now allow us to deliver tools of molecular biology to crops. So this is specifically going to be DNA, RNA, or proteins, which are molecules that are needed to be introduced into plants for their genetic transformation. And that's kind of step one. Step two, of course, is then taking those transformed tissues and regenerating them into crops that can propagate these beneficial traits. So towards that goal, we look at a few features, the first being a platform that could be plant species independent, which tends to favor abiotic methods that have not necessarily co-evolved to transfect or to transfer genes from one organism to another. And then non-pathogenic and non-integrating are key traits that are mostly perhaps regulatory in nature in terms of their benefits, where a non-integrating platform where the DNA that's being introduced into a new species inherently does not integrate could avoid the necessity to then cross out any transgenes that are introduced through conventional methods. So current tools that exist for plant transformation, these are two of the gold standards. The first is Agrobacterium mediated delivery. Agrobacterium is a plant pathogen that has evolved to transfer genes into plant tissues of select species, and as scientists, we have been able to engineer strains of Agrobacterium to do so across a broader range of plant hosts. Agrobacterium classically does this by introducing this foreign DNA into plants and then integrating this DNA into the host genome, although strains of Agro have been engineered to do so in a non-integrating fashion. The second approach is ballistic particle delivery. So ballistic particle delivery involves dehydration of DNA or RNA or proteins onto the surface of gold particles that are then shot into plant tissue using a high-pressure gene gun. And this allows disruption of the plant cell wall, cell membrane to introduce these foreign biomolecules for the intended genetic engineering applications. For both of these approaches, there's a lack of control over the frequency of transgene integration, especially in the case of biolistics, and the location, which can result in endogenous gene disruption if the integration happens within a gene cluster that's important for the fitness of that plant species. Now, if we're thinking about how to introduce foreign matter into plant cells, it's important to think about some of the physics behind this. And thinking about what makes plant cells unique relative to animal cells or traditional mammalian cell culture, where methods like electroporation and heat shocking are fairly common, we look at size exclusion limits of these various membranes. So if we're only considering the plant cell membrane, the lipid bilayer as the predominant barrier, which is the case for most cell types, here the size exclusion limit is on the order of about 500 nanometers. This is about as large as something can be before it no longer translocates along that particular membrane. The plant cell wall has a size exclusion limit estimated by calculation between 5 and 20 nanometers. And this is, again, a very broad estimate that's likely species-dependent, tissue-dependent, and age-dependent. But as you can see, it will pose the dominant barrier to exogenous biomolecule delivery, especially if we do not want to use force for the introduction of foreign matter. And so this brings about the concept of nanotechnology as a class of material science that will enable the synthesis of materials within these size scales. So not only the dimensionality or the size of these materials becomes important, but also their constituent materials. So here I'm showing different dimensionalities of carbon-based materials where we can go from 0 dimensions to 1 dimension to 2 dimension, where all of these materials have different properties, and specifically with regards to their delivery potential, the surface area to volume ratio becomes important. So keeping at least 1 dimension below the size exclusion limit of the plant cell wall in the case of 0 or 1-dimensional materials, but also having enough surface area onto which we can graft biomolecules and do appropriate surface chemistry, leaving the only option being the 1-dimensional nanomaterial case. So the concept that my lab explored here was one in which we would be able to graft biomolecules onto the surface of these single-walled carbon nanotubes, that they might translocate across both the pores of the cell wall and the cell membrane, and be able to deliver cargo to the nucleus in the case of gene delivery, although we can also deliver RNA to the cytoplasm for RNAi different applications. Here, the concept would be that we could do the chemistry in a way that these plasmids would be accessible for transcription and translation without premature desorption prior to getting to the nuclear destination. So for these studies, we're using a green fluorescent protein reporter gene for our preliminary data sets where we can deliver these reporter plasmids with carbon nanotubes to see how the efficiency varies with different surface chemistries. We can test these in different plant varietals to see if we can get expression across different species. Not all of these have been tested in my lab. I'm just going to show the results that have been. The concept here is we infiltrate these particles along with the genes into plant tissues, and then with confocal microscopy and molecular methods, see if we can get GFP expression. So as shown here, we can see fluorescent protein expression in a standard model plant such as Benthamina, and also non-model monocots and dicots, arugula, wheat, and corn. Some of the data on the left shows the variability with regards to surface chemistry. The takeaway here is that plasmids that are adsorbed very tightly to the nanoparticle surface are most efficient at being able to drive GFP expression in these tissues. One of the interesting features of using nanotechnology is the level of control that we can have over how tightly these plasmids are grafted, and therefore putatively how well they can be accessed by the endogenous machinery of the plant cell. One of the features of the platform that we observed was the transience of the expression, where we would see GFP expression usually peak at day 2 or 3 and subside by day 10, which we can confirm with molecular methods such as Western blotting and also quantitative PCR of the GFP mRNA. What this suggests is that there's a lack of integration of the transgene, and this opens up possibilities for using abiotic technologies for the delivery of DNA, RNA, and protein to plants in a way that could help target transgenes or genetic modification of these crops for food, bioenergy, and even medicinal applications. Towards this end, I see a lot of potential in incorporating genome editing technologies in this space, specifically for editing of crops that can only be vegetatively propagated. And the concept here being that the ability to transiently deliver a transgene and transiently express a nuclease such as CRISPR-Cas9 would be able to modify the genome of a plant, especially if that modification is a knockout of a gene, where that knockout could be inherited by that plant tissue, but the transience of the expression and the non-integrating nature of the plasmid would avoid the need to use segregation-based breeding to cross out integrated transgenes in future generations. Towards this end, we've started doing tests with CRISPR plasmid delivery in the lab, using first just guide RNA delivery to transgenic Cas9 expressing plants. Our platform, both with GFP expression and CRISPR, is about half as efficient as Agrobacterium, which is not too surprising to me given, again, the lack of constitutive expression of these nucleases, but it's something that we're working on not just in leaves, but also in moving to other tissue types that would allow us to do so in regenerable tissues for heritable traits that could be conferred through CRISPR. So with that, a brief summary of technologies that have been developed recently in my group for DNA delivery, RNA delivery, and hopefully soon protein delivery as well, and hopefully motivating the conversation over what constitutes genetic modification, regulatory oversight and public acceptance of GM foods, and also how nanotechnology will play in this very quickly evolving space. Thank you.